Hello and welcome to the last day of MD and DI's Focus on Fundamentals course, keeping your biocompatibility program current in a changing regulatory landscape, sponsored by Nelson Laboratories. I'm Chris Keach and I'll be your moderator today. We have just a few brief announcements before we begin. First, this webinar is designed to be interactive. The dock of widgets at the bottom of your screen will allow you to learn about today's speakers, download resources, share this webinar via social media outlets, and participate in our Q&A session that takes place at the end of today's presentation. Also, just please note that we'll try to get to as many questions as we can in the time that we have left at the end of today's program, but if we're not able to get to your question today, someone will get back to you after the program is over. The slides will advance automatically throughout the event, and you may also download a copy of the slides by visiting the Resources widget. Towards the end of our webinar, we will ask you to complete our survey found on the right-hand side of your screen. Please take a moment to fill this out before leaving us today, as your feedback will provide us with valuable information on how we can improve future events. And lastly, if you're experiencing any technical problems, please click on the Help widget found at the bottom of your screen or type your issue into the Q&A text area, and we will be glad to offer one-on-one assistance. And now, on to today's class, how to address regulatory change in your current biocompatibility program. Discussing today's topic from Nelson Laboratories is Audrey Turley, Technical Consultant. To learn more about our speaker, please visit the Bios widget. It's with great pleasure I now turn this special session over to Audrey to begin. Audrey, take it away.
Thank you, Chris, for that introduction. So thank you all to joining us today for our Day 3 webinar. So I just want to recap. The first day was presented by Thor Rollins, who discussed what was actually changing. He presented several standards and discussed the changes also in the MDRs. And then Day 2, we had Drs. Jorgensen and Vertomen, who discussed chemical characterization testing. That's a large area of change, and so it did take the full day. That was a great presentation. So today we're going to cover what to do with all of this information now that you have that. How do you translate this into your own biocompatibility program? So that will be the focus of our conversation today. So I wanted to start out reminding ourselves that even though there is a lot changing as far as wording and direction in individual standards in the 1093 suite, we're still focusing on risk. So even amongst all the change, our overall goal is to ensure that we are staying focused on assessing the risk of a medical device from the materials and manufacturing process. This is not changing or going away. Assessing risk can make biocompatibility more complex. However, this direction allows us to use data that already exists for a device or compound. So, if you're changing something on your device, we can use data you already have to support that change, data that's already published in the industry, clinical use information when you're just moving to a new market. So the goal here is to use this risk to make sure we're making scientifically sound decisions, and in the end we can reduce cost, time, and animal lives necessary to bring a product to market and keep it on the market. So I always like to bring up the 10993 Part 1 title, which reminds us that our overall, this is the umbrella standard that we go to for biocompatibility, and it states that this is an evaluation and testing within a risk management process. FDA's guidance document also has a 9-page section titled Risk Management for Biocompatibility Evaluations. This section is really nice because it provides us with a line of thinking from the US FDA on how to identify risks in medical devices. And then we have pages of examples on how to use data we already have and assess the risk using existing data, literature review, clinical history of use, and the list goes on. So although this is from the FDA, from the US FDA, the line of thinking can be broadly applied in the industry for other markets. So this is a nice place to start if you need some help getting going. So I'd like to bring up this first paragraph, one of the introductory paragraphs to this section within the FDA's guidance document. They identify what we should assess or what we should be considering when we are assessing risk. So they say material components, our manufacturing processes, clinical use of the device. Then we can take those three components and look at that from a biocompatibility perspective. And then we can address the identified risks either through testing or other evaluations. So this allows us to do some written risk assessments, use literature research, and possibly some chemistry testing as well. This definition of risk is one I go to when I talk about it here in this context. So we use from ISO 14971 that our definition is the combination of the probability of occurrence of harm and the severity of that harm. So when we break this down into layman's term, how likely is the harm to happen? How frequently can it happen? And if it does happen, how severe will that be to the patient? So this is a presentation overview. I really broke this down into a few categories when we talk about how are we going to assess change. So, Thor presented all of these changes that are happening in the standards, and then Drs. Jorgensen and Vertomen focused on just the extractable leachables testing. That's a large area of change. And so translating all of that change to, okay, what will that look like as far as projects and maybe written assessments that will happen within your facility. I've broke that out into 4 areas where we will talk about material characterization. And this is taking a different turn on addressing material characterization from what Drs. Jorgensen and Vertomen presented yesterday for day 2. So this, if you listened yesterday and were maybe a little concerned about what you will need now to be compliant with the 10993 standard that was recently released, I hope to put some of your questions, address some of those questions, and maybe help some of you not be as nervous about what's needed down the road. And then we'll talk about gap assessments. This is going to be a large part of adjusting to the new MDRs, where they are asking for gap analyses now to understand how your medical device is now compliant with the new MDRs. So we will go through that extensively. Then we'll talk about a biological evaluation report. So this is not new. This is a requirement that was laid out in the previous version of 10993 Part 1, specifically in Clause 7. However, I want to let you know what we're seeing from feedback from notified bodies and regulatory bodies and how they are expecting those to look and what information they're asking to be included there. And then biological evaluation plans. So this is an initial risk assessment that can help to address new standards that are coming out, and also, especially with the MDRs, if device classification has changed and new testing requirements are needed to be followed. And this is a great initial risk assessment to justify how you're going to follow those new requirements. So let's start with material characterization and really try to define that. I feel like this is a very vague line of testing. So in the new 10993 Part 1, if you go to the table where they're asking for biological endpoints of concern, the table we would frequently go to reference to understand, what are our risks of our device, you'll see that material characterization is required for every device. However, the guidance on what that looks like is very vague and open to a lot of interpretation, which can be good, but can also be dangerous if we set the bar inappropriately, be a little too high inappropriately. So this definition is taken from 10993 Part 1, and this was the draft that was issued at the end of last year before it was circulated. So because it recently got published, this part got published after I had submitted my slides for the presentation. My slides are already outdated to an extent because it's just such a rapidly changing field right now. But this definition still applies. So we're defining material characterization as the broad and general process by which a material's chemistry, structure, and properties are evaluated and measured to include assessment of compositional, structural, and mechanical properties if appropriate. So those last 2 words that I've bolded in my slide here really let us know that the door is wide open for how we are interpreting what a material characterization program should look like for every single medical device manufacturer. So how do we make that decision? Do we always need to do extractable and leachables testing? Is there some other version that we can do that keeps our testing burden appropriate based on the device type? So that's what I'm going to focus this beginning section of my presentation on, is what do you do when you don't do extractable leachables testing? So let's talk about what it should include outside of just testing, some of the information that is needed. This quote was taken from 10993 Part 1. So we always start with, Part 1. We understand where we need to go after that. And in Part 1, it is specified that you need to identify why you chose your certain material for your medical device. Now, this decision is made very early on in the manufacturing process and typically comes from the engineers. Understanding the materials, the properties, what the device is intended to do, and also common materials within a specific industry. So all of those things are taken into consideration. So that should be documented. And so there's a thought process here where we need to consider, how are you documenting this type of information at your own facility? There isn't a hard line of what it should look like, so that leaves you some flexibility to incorporate into the system you already have. So keep it with your device history files, and if you need to document it and you don't have this in one location, think about how you can pull that together. So as a laboratory, this is not something we would be able to pull together because so much of this information is happening and being decided within the manufacturer facility. So this should be a separate part of your material characterization, but also an important part that's documented. Okay, so this next slide is where we go to then after we leave 10993 Part 1. So 10993 Part 1 says, get the information on why you chose the material, and then do material characterization according to Part 18. So if we go over to 10993 Part 18, which is also just recently, a new revision was since I have submitted these slides for the presentation, but this line of thinking is still consistent with the new version. It states the extent of chemical characterization required should reflect the nature and duration of the clinical exposure. So I want to focus on that for a minute. This allows us to determine, okay, what's our contact with the patient, what's our risk, and how far do we need to assess that? So right here is where we are going to decide if we do extractable and leachables testing, or if we can do a literature review, or if we have good supplier information on this material that we're choosing to use, and how invasive or how extreme is our manufacturing process? How many compounds are we using? Do we do a cleaning validation? You can see that you kind of go in many different directions when we're looking at material characterization. So I would really like to say that this needs to be done very carefully because we want to set the bar appropriately. And what I mean by that is, we're in an industry of overachievers. We really like to set the bar high, and that's what drives our industry. But I want to make sure that we are doing this appropriately. So we don't want to start doing material characterization on everything that increases the testing burden to an extreme level. And then what does that data provide in the end? So if we have a surface contact device for a few minutes with a patient, do we need to do full extractable leachables testing on that surface device versus a permanent implant that goes through some extensive manufacturing? So those are two very large extremes in what we're looking at, but that is how diverse the medical device industry is. So we want to be careful that we're not stating for our own biocompatibility programs that we're going to do extractable leachables testing on everything because it's a medical device. We're going to develop an appropriate material characterization program based on the nature and duration of our device contact. And you should consider each material. This gives you the opportunity here to say, okay, this is a titanium material that's made according to a standard that is specified for this type of application. So you can really eliminate a lot of testing, a lot of burden by stating that information. So this leads us into our next slide where we want to talk about material information from your supplier. So a lot of times, especially in our field, we love it when we see these certificates from suppliers that say, okay, we've done USP Class 6 testing and we are ISO 10993 compliant. So we really like that information. They'll say that they're medical grade. Some claims that I've seen that are not appropriate, and I also want to take a minute to just give a voice of warning. If a supplier states that a material is FDA approved, there needs to be some careful understanding of that statement. FDA specifically states in their guidance documents that they do not approve materials. Now, a material supplier may have submitted like a master file, but that does not mean that their product is FDA approved. That means that they've done the appropriate testing, they filed it with the FDA. So when you submit your product and say you've used this material, FDA can then open that master file and access the information that the supplier has submitted. However, the one downside to that is, as a user of that material, you do not have access to the master file. So unless the supplier gives you that information and tells you what's in there, you're never quite sure how that correlates to the use of your device. So just a voice of caution there to have a better conversation with your suppliers and understand what they're including if they've submitted a master file, if they're claiming FDA approval. Okay, so my voice of warning there. Let's talk about what USP Class 6 testing is so we can understand that from a material perspective. So Class 6 testing is clearly from USP. This was originally used for testing pharmaceutical containers so that they could ensure that nothing was transferring from the container into the drug that would be delivered to the patient. So this testing regimen consists of irritation testing, systemic injection, and a 1-week implantation. So if you're familiar with the 10993 standards, these 3 tests do not fully address all the risks for any one categorization of medical device. It does let you know that you're starting with a material that shouldn't add anything to your end biocompatibility. However, this doesn't take into account your manufacturing processes and your sterilization. And we're still missing critical endpoints like cytotoxicity and sensitization. So although this isn't a great plug to eliminate all your biocompatibility testing or say that, hey, it's USP, so now we don't have to do anything, it lets you know you're starting with a good material and should be part of your material characterization so that you keep notes within your company of what you're expecting for these raw materials. Say everything has to be USP Class 6 or 10993 compliant. It gives you a good place to start. So let's talk about 10993 compliance. So I get a little riled up when I see this on certificates because I'm not sure how you can be 10993 compliant for a raw material. A raw material that you're not sure how it's going to be used. Compliance to 10993 standards is based on your type of contact and your duration of contact with the medical device. So, if you're sure that your raw material is only used for a specific application, you can state that. But more importantly, we need to understand what tests were actually performed so you can determine how relevant that is to the use of your final finished device. And also what's nice to understand is if they did any of the testing on the raw material post a sterilization procedure, you can understand if your material, that material is compatible with the sterilization cycle or the sterilization process that you choose. So that's really all that I was going to say about material characterization. mainly I just wanted to open the door and let you know that there are many different ways to approach material characterization. Don't necessarily go down the tunnel of extractable and leachable testing if it's not appropriate. If you have a long-term contact device, material or the extractable leachables testing is excellent for that. I highly recommend going to the US FDA guidance document where they have outlined exactly how to apply that extractable and leachables testing to your full biocompatibility program. That does result in reduced biocompatibility testing. I know that Dr. Jorgensen talked about that yesterday too. So I don't want to beat a dead horse with the material characterization, so let's move on to gap assessment. We're going to spend a little bit more time here. So gap assessments are really done to determine if your previous biocompatibility testing is applicable. So in our gap assessments, our very first one of our very first sections is to really outline what we're going to do. So we're going to review what was performed and compare it to the current standards. So this also includes addressing standards that have been developed, like the MDRs as well, visions of an existing biocompatibility program, and then seeing if the testing was performed appropriately. So we're going to step through this one step at a time on how I perform these. And I wanted to give some examples of what I've come across where this has proved really valuable, and also to kind of make sure, just a little bit of warning to make sure that your biocompatibility programs in-house are being run appropriately and that your staff is appropriately trained to handle, review the. So the first thing I'll do is I'll look at the previous testing that was done and compare it to what I know is required in the standards. So one example is I reviewed a sensitization test. So this is a combination of things that I've seen. This isn't specifically something I've done for a particular client, but a variation of what I have seen. So sensitization testing was done on 10 animals. Sensitization testing is run on a polar and a nonpolar solvent. And so for this particular example, we're going to say that 2 animals died, 1 from each. So although, the laboratory said, hey, these deaths were not related to the device, they did an autopsy and they were able to see the animal had some complications anatomically, actually, of them, that were not detected prior to starting the study. However, now we do not have the appropriate number of data points. If it had just been 1 solvent, we could maybe have addressed that if it was just 1 animal with just a risk assessment on that point. But because 1 died from each solvent, we're now down 2 data points. You really need to have 10 for it to be statistically appropriate. And based on the guidance document, now no longer meet the guidance document. So we did recommend that additional testing should be performed, or a risk assessment based on previous clinical data, or some additional chemistry testing, if apply that to some sensitization, predict sensitization response. Okay, so the next one, we'll compare, the current standards in the guidance document to the full program. So not just the details in the tests, but what tests were selected and what were performed. So if you've looked at the table in 10993-1, you'll notice that it'll have, these biological endpoints. It lists them by, it's a general category: cytotoxicity, sensitization, irritation. We go through it like that. But one thing to remember, if you go into each of those individual standards, there are multiple options for each test, and sometimes there are multiple tests required to meet that biological endpoint. And genotoxicity is one of those areas. And it seems that is not clearly understood, and I've experienced and seen many, many times where the full program wasn't completed. So just in this case, just an Ames test was performed. That's one everyone will typically do because it is cheap, it is quick. As far as genotoxicity goes, it's one of the cheaper tests, and it is fairly quick. However, Ames testing is run on bacterial cells. and the regulatory agencies really want to see some results on a mammalian cell line. And so they ask for you to do the AIMS because that's a great beginner point, and the type of mutation that looks for is very informational. But they also want you to do testing on the mammalian cell line, number one, because it's mammalian, number two, because it does look at a different type of mutation. So in this case, I would recommend complete their genotoxicity battery by performing one of the mammalian tests, or look and see if they've done some chemical relate those results through a toxicological risk assessment to address genotoxicity. So once we identify the gap, we will recommend the testing, but that recommendation depends specifically on the unique situation of the manufacturer, where we want to try to use the information they already have available. We're always keeping in mind that we are assessing this based on risk. We don't want to just say, go and do a test so you can check off the box. Make good decisions about the way we're addressing these gaps. So one of the biggest things that I see in these gap assessments is the risks that are identified for a specific device. So when we talk about risks, we're really talking about identifying the right biological endpoints according to 9.3 Part 1. So here we have an example with a device that has limited tissue contact. So the company did cytotoxicity, sensitization, and that's a basic requirement in ISO 10993 Part 1. They've submitted this to the FDA, that the FDA has asked for this type of device contact to address acute systemic and material-mediated pyrogen risks as well. So our recommendation then is to follow up this gap assessment with either a written assessment, if you have enough clinical history information where we can pinpoint these specific side effects with a patient to understand that this is not a risk, or to go ahead and to complete the test as FDA would request upon. Oh, I have this in here as an extra slide. Oh, here we go. So the conclusion here, what we want to say is that based on the review of tests performed and the current standards, the biocompatibility of the device is well supported. And we'll typically follow that up by saying additional animal testing is not recommended, would not provide useful data, so that we're sure we're saying we don't think anything further needs to be done. So you can see there's very different situations that have occurred for every single device that we've reviewed with these gap assessments, and it does depend too on the market that we're moving to and where they originally submitted and how long ago that was performed. Some of these devices that have been on the market for years, just did maybe the big three: cytotoxicity, sensitization, and irritation testing. And although there's many, many years of clinical history of use, sometimes the problem in applying that data is that we cannot pinpoint it to the specific risk that the FDA or regulatory agency about, like an acute systemic toxicity. Depending on the environment and use of the device, that's very hard to target to one single device. So they're wanting to have more detailed information about these devices, identify if we've let things slide over decades of use that hasn't been captured for a device. Okay, so now we're going to move on to a biological evaluation. So the big question here is, was the testing reviewed by an expert? And this comes specifically from Clause 7 in ISO 10993 Part 1, where they outline, what's required when you review these reports and how you should understand it from a bigger picture perspective as well. So this is really wordy. I just wanted to put this on here so you guys could go to your 10993 standard and review that and make sure that your biocompatibility program is including this final portion. We have had to submit our CVs, written biological evaluation reports for notified clients submitting notified bodies, and we have had many clients come to us after their submission been rejected by a notified body because they do not have a summary report. So let's just go through these a little bit at a time. So I've just highlighted and marked. Where the what's important from each standpoint. So, your strategy of the program really comes from your initial risk assessment. Are you looking at the appropriate risks of the device? So that needs to be addressed again in your summary report just as another justification. So the way I like to think about this biological evaluation report is that this is the conclusion in your story. So we want to have a good ending, and in that conclusion, we're kind of summarizing everything that's happened. We do need to state our beginning place of thought because that's going to tie into the end where we're saying that what we did was appropriate. So again, here in Part B of this, we talk about the acceptability of the material. Again, this is coming from what you're documenting in-house and should be captured somewhere in the design history file. And then the adequacy of the material characterization. So this is how well you established and defended your approach to how you characterized your material. So that conversation is important to have up front. And again, to reiterate here at the end when we state all the testing that was done. Rationale and selection for waiving of tests. We want to go through, again, we want to make sure we're not just checkboxing. We need to state why the tests are appropriate, and then really, really be specific on how the device contacts the patient. One thing I've noticed a lot in submissions is that there's a lot of conversation about how great this device is and how all the gadgets and components and everything that it does, but there's not enough conversation of how it's contacting the actual patient or fluids. So enough good discussion there. Interpretation of existing data. So this is where reviewing the reports and understanding what the standards require. There was one evaluation report that I was writing where we had another issue with an animal death. And so again, even though the death wasn't related to the animal, we still did not meet the requirements in the standard. And so some additional risk assessment needed to be performed to address that death and more conversation with the laboratory about the death of the animal. So I would say if you have staff at your facility that are reviewing these reports, you can invest in their training and development so that they can understand not just look for that conclusion and rely fully on the laboratory, because the laboratory's perspective of what's okay versus what's going to be okay for your submission might be two different things. So we want to make sure that for your submission, everything is covered. And then the need for any additional data. So what we've seen often, we'll go through and be doing this testing with clients and they call us because something has happened. Maybe a failure has happened. Maybe something was identified in the chemistry testing. We need to do some more targeted analysis. And so this is where we will have that conversation. So this is a discussion that is happening throughout the testing phase, but it needs to be explained here in the biological evaluation report so that there's an understanding of why the additional testing was done and if the overall risk was eventually mitigated. And then at the end, we're going to make an overall conclusion about the biological safety of the medical device and putting everything together to determine if all of the risks were addressed and if the data is appropriate and accurate. So when we talk about the strategy and program content for the biological evaluation of a medical device, did you follow the appropriate standards for testing and test selection? Are all the risks identified for the device? We want to make sure we're stepping outside of just the 10993 standards and understanding if there are any vertical standards. So when we think about 10993, all of those standards together, those are a broad application to the medical device industry. And we know this now that many, many risks of devices are not being assessed for their specific use. So standards are coming out, we call those vertical standards, where they go more in depth based on the application of the device. We see this for breast implants, for dental devices, and for breathing gas pathways, just to name a few, that we want to make sure we're following all the standards appropriate for the specific device and not just 10993. Part B, the criteria for determining the acceptability of the material for the intended purpose. Again, this is determined by your manufacturer. Also information to include in your material characterization assessment. The adequacy of the material characterization. So again, referencing 10993 Part 18, the U.S. FDA guidance document, and justifying and rationalizing an appropriate program for your device contact and duration. And the rationale for selection and or waiving of tests. One thing I want to talk about here is that just because a test has been marked as, hey, this is a risk of concern in 10993 Part 1, we want to make sure that we can actually test the device in that test system. We've had that be a problem where maybe, let's say, like a gel product that would be applied frequently or at a certain location would be marked for implantation. However, that's not appropriate. We would have a very difficult time actually performing an implantation study because the gel would not stay in place. So when you're developing your initial risk assessment, you want to make sure you're also taking into account, can we actually assess this endpoint for the device? And if not, what are some alternatives to address the actual concern here for use of the device? And again, the interpretation of existing data. So please review the full report, not just the conclusion. And some areas of concern that the notified bodies and regulatory agencies are looking at: extraction conditions, extraction appearance, and was the study criteria met? So we really focus a lot on extraction conditions because we want to make sure they were done at a high enough temperature for a long enough duration. And then extraction appearance. So make sure that your laboratory is giving you that data in your final report. It is specified in the 10993 standards that that should be explicitly recorded. And if it's not, the testing will need to be repeated just so that can be verified. And we have seen testing rejected for not having this information documented. And the need for any additional data to complete the biological evaluation. So we talk about DHP. That's a topic that's very hot right now that needs to be addressed. So do you need to include some additional targeted testing for labeling? Do you have special populations, a special target organ that needs to be addressed as well, especially if you're doing neural application devices? The data for that type of contact is very limited. So it becomes a very customized project that we end up developing. So we want to make sure we've addressed all of that and discuss it here in the biological evaluation report as well. And then the overall, our overall conclusion. So a lot of things that we'll deal with at this point is maybe we saw cytotoxicity failure, but everything else passed. Can we still claim that it's biocompatible? And we'll add a risk assessment section into the biological evaluation report where we can address that. And then, as happens often in the medical device industry, the material and manufacturers can change after you've already done your testing. So how do we assess that change? And we can do that right here in the biological evaluation report. We want to make sure we're putting that all up front in one place to have a good conversation. And again, addressing, possibly slight irritation responses. We can address that as well. Just really looking at the risk of the device and the way it's used. So that wraps up for the biological evaluation report. One thing I will say is, in the past, I don't think that I know that they weren't as required as they are now, but we have seen that the regulatory agencies are not wanting to come back and make all these conclusions. They want to hear your line of thinking. They want to see how you've addressed the risk, and they're giving you the opportunity to have a voice. So this is a great chance to use that and to help drive the expectations appropriate for what we can actually do and where the risks are with the patient. And so now I'm just going to talk briefly about biological evaluation plans, how to address new standards. So I want to step back and look at a big picture for your biological safety evaluation programs. So this is, our overall view. This is really how the program should be designed according to the standards. So we start in phase 1 with the biological evaluation plan, where you're identifying your risks, you're justifying what tests you're doing and not doing, how your testing is going to be performed, extraction parameters, everything we talked about that are important for a biological evaluation report. We're going to talk about that up front with justifications and rationale in our initial risk assessment. And then we do the testing, address any problems, do additional targeted testing, and then finish that up with a biological evaluation report. So when new standards come out, the best thing to do really is to address how you're going to approach the testing, and that can be done in a BEP. These BEPs are also really nice to aid in initial conversations with regulatory agencies like the FDA. They specifically asked for that in their guidance document. So we still start with identifying our risks from what we already know. So this is a flow chart that's from 10993 Part 1. However, in some of the new standards that have come out, like 18562, they also have a similar flow chart to help guide your conversation about identifying your risks. So always start with what you already know, and then decide, okay, how much testing is needed? What do we know about our materials already based on the risks that are identified in this new standard? So we go into material characterization, just what we talked about in the first section of the presentation. So I'm putting it here again so you understand the context of the conversation is happening in this initial risk assessment. So these are the same quotes, but just to drive this when you're looking at the presentation when I'm not available, is that you understand we're talking about material characterization right from the beginning. It's driving what you decide to do as far as testing. Another thing to talk about is, what are you going to test in your initial, with your biological evaluation plan when you look at these new standards. So we have a raw material to a finished device, and we have all the processes and sterilization in between. We want to make sure that we're including those steps, and if you can document that your processes and your sterilization are not affecting your raw material, either because you have testing that supports that from your raw material supplier, so nothing you're adding is going to change it, then you just have a very detailed conversation. Saying it is the same is not enough. It needs to be lined up in a table, going through each processing step, and then understanding your raw material as well. And so 10993 Part 1 still states that testing does need to be performed on the sterile, if it's offered sterile, final product. So if your device isn't sterilized as part of its manufacturing process, then you don't have to sterilize it. But if it is, that should be considered in the testing program. Some other things to include in the biological evaluation plan is how we're preparing the test sample. So do you have electronic components in your device? Did you exclude those? Have a family of devices, so you developed a master product? Where you made a different product to represent each thing? Do you have multiple devices that are from one family and you group them together? Did you have to manufacture a coupon to represent all your materials? This is especially applicable for implant testing. So these are the questions that we talk about and address when we do sample preparation in our initial risk. I have this extra slide in here. I just wanted to go over it again a couple of times with you guys, I guess. Okay, here we go for the extraction time and temperature. This is also important because the FDA specifically has asked certain parameters based on the contact duration of the patient, of the device with the patient. So typically this first one here is for cytotoxicity only. Now this is all listed in 10993 Part 12. However, it doesn't give a lot of requirements on when each extraction time and temperature can be used or should be used, just that it needs to be justified. So cytotoxicity is typically done for 24 hours. However, the FDA has started to ask for implants that cytotoxicity be done for 72 hours. So that's just one of those nuances that needs to be identified up front so that the testing is performed appropriately to the device. Typically, we sit here with the 50 degrees for 72 hours. That's most common. So again, just keeping all of this information in mind to make sure that you're documenting that in your initial risk assessment. And so here's just a summary and kind of a flow of how we address our biological evaluation plans. Again, starting at the front with material characterization, because that does dictate how you approach the testing. And then again, just talking about your device, ensuring that you describe how it contacts the patient, include pictures. That's always helpful. It's important to remember that when you submit to the agency, they will divide up your file to the appropriate experts, so they're not always seeing all the information up front. We want to make sure that we give them what they need in one document, so if it gets sent to their biocompatibility expert, they have all the information that they need right here. So just as a summary for my presentation today, this is what we talked about for material characterization, how much research and testing is appropriate. This is really based on your contact type and duration. Gap assessments, have your testing reviewed by an expert, that can understand the nuances of the test and also understand the standards that are current now and to make sure that they relate. And then biological evaluation report, ensure that a BER is written because rejections will happen without summary and good conclusion. And then a biological evaluation plan, have an initial risk assessment in place so that, it also helps develop your biocompatibility program for your company so that there's some justification or documentation for the line of thinking when you did the biocompatibility for that particular device. So that's it for me. I'm going to turn the time back over to Chris. I'm going to open it up for questions, I believe.
All right. Well, thank you, Audrey, for a great presentation. And now, before we begin with today's Q&A, please direct your attention to our webinar survey, available on the right-hand side of your presentation window. If you closed the survey, you can reopen the widget by clicking on the icon along the bottom of your screen. Thank you in advance for filling out the feedback form. Your participation in this survey allows us to better serve you. We're now going to move on to the question and answer portion of our event. As a reminder, to participate in our Q&A session, just type your question into the text box located to the right of the presentation window, or click on the Q&A icon at the bottom of your screen. And please note that we're going to try to get to as many questions as we can with the time that we have left. If we're not able to get to your question today, someone will get back to you after the program is over. And joining us from day 1 and day 2 will be Thor and Annalise. They'll be joining Audrey for today's Q&A. So let's get started with our first question. And let's go down here and find it here. Hold on one sec. There we go. Okay, so our first question is, what qualifications should an expert reviewer have?
That's a great question. This type of science isn't taught in school. You can't get a degree in biocompatibility. So when you are determined to be an expert, it's based on your experience in the industry. For us here at the lab, it's also based on our participation in the committees that help write the standards. So those two combined, of course, with an appropriate degree or scientific educational background as well. So I would say it's like the 3 pillars there that help determine if you are an appropriate expert. And again, like I've said, we do typically submit our CV when we submit these BERs because they have been requested in order to determine that.
All right, thank you, Audrey. I have another question here for you. Do you find it to be common to test aged products?
That is a question we frequently get. So we can and have in the past test aged products. It's not required anywhere to do that. So what we want to focus on is if we know that the material is going to degrade or be stable over time in its condition, packaging, and shelf life. So if there's a concern about that or not enough information known about the material and its stability, we could do some chemistry testing and then do some comparative biocompatibility where we wouldn't necessarily want to do two sets of biocompatibility on a freshly produced and on an aged product. Again, just trying to make sure we assess that risk appropriately but find the balance of testing burden on the manufacturers. So yes, we do test that. I wouldn't say it's common or frequent, but it is something we do assess when a client's request it.
All right, thank you, Audrey. Our next question here says, if you have an existing or marked product for which biocompatibility testing is completed but a BEP was never done, how do you recommend catching up to the requirement to have a BEP?
That's a great question. We've actually done BEPs after the testing was performed. One thing that could be done is a brief memo that could be written, and then if you have a really nicely documented BER, that is a way to tie those in together. And so if you aren't in a place where you want to do these BEPs, I would make sure you have a nice BER with all the details necessary, and then maybe have a memo, a memo to that file that adds some information that might be missing from your summary report.
All right. Thank you, Audrey. Another question here for you says, how should you assess a component chance in your medical device?
So this is Thor, and I believe that probably would be change, I'm hoping. So for me, the way that we approach a change is really just the risk of that change. So we look at the surface area of the component that's changing versus the surface area of the overall device. And then we also look at the materials and the processes that are changing. Oftentimes, we can address those changes by using some simplistic chemistry tests to evaluate the impact of the change. But remember, dose makes the poison. So the more of a device that you're changing as far as surface area goes, the bigger the risk to the patient and the bigger risk to the testing itself. So you first want to look at the impact from a surface area of the change. Then you look at the materials and the processing, like you would with any biocompatibility risk assessment. And then ultimately, you want to try to find a way to mitigate that risk, and that could be through limited testing, or it could have to be repeating biocompatibility based off the change itself.
All right, thank you, Thor. Our next question here says, can you speak to how the new MDR will impact the need for testing and gap assessments, specifically around the level that the clinical history will be enough?
Yeah, this is a great question, and honestly, we could do a whole webinar just on this question. So with the new MDRs coming out, there's still some uncertainty right now in the industry regarding the impact and if the time that 2020 will be enough time to do this. But yes, it's going to impact a lot of the testing. In fact, we have quite a bit of customers right now that are literally having us go through products that are existing on the market. We try to do as much of the market research as possible to try to get a sense that does play a role in your risk assessment. But unfortunately, a lot of times that information is not sufficient because there's so many things being used and then during a procedure that it's hard to trace any kind of reliable information. And then there's a lot of endpoints from a biological status like sensitization, irritation that may not be recorded in some kind of follow-up. So in reality, what you have to do is go through products that have CE mark that you're going to have to resubmit to your notified body, reevaluate the gaps that they have to the current testing standard, fill in those gaps. A lot of times we use chemistry to help fill in those so we're not doing unnecessary animal tests, along with a risk-based approach with materials, very similar to what Audrey talked about with the BEP. And then we do this kind of gap to evaluate what may be missing. So it's actually quite a bit of work, and it's something that, like I said, a lot of companies have already started. We've worked with quite a few already, and we've actually partnered a little bit with TÜV SÜD to try to understand more about the impact of companies and what could be looking down the road. So all of this plays a role into this kind of assessment.
All right, thank you, Thor. Another question here says, should chemical characterization of leachable and extractables be conducted before an expert toxicologist is commissioned to perform biological evaluation plans, or BEPs?
Hello, this is Annelies. I will take this question. So generally, this is the chemical characterization is also described in the BEP, because in your BEP you will state why you are going to use chemical characterization, which test it can replace. So and afterwards, so the BEP is written, then you do your chemical characterization, and then you do your tox assessments. Then the results of these tox assessments will then end up in the biological evaluation report.
All right, thank you, Annelies. Our next question here says, what kinds of materials would actually fail an acute systemic toxicity test?
So this is Thor again, and this is actually a very interesting question. So as I've kind of mentioned on day one, the most sensitive test we have is cytotoxicity, and so there are, when you look at a failure, that's mostly what you see. So in reality, we don't see the acute systemic toxicity fail very often. And when you look from a toxicity standpoint, very few compounds are extremely acutely toxic. So as soon as they're exposed to the animals, there's a reaction. Usually it takes a repeated dose or a buildup to reach some kind of level in the body that would then cause that whatever toxicity reaction would happen. There are some, however, that would cause it, and I know some of these compounds are scary, but for example, there are some plastics that once they get to a certain temperature could actually release some cyanide, which obviously you would not want to inject into anybody. So in that case, it's very, very rare, but we have seen every once in a while in the last 17 years, a compound that is injected into these mice that would have an immediate reaction. And then those are the kind of compounds that we're automatically looking for that would elicit it. So that's why the acute systemic toxicity is in place, because it's not so much, I want to say, material, but material byproducts or from processing or residuals or misuse of the material that would cause a reaction to occur.
All right, thank you, Thor. Our next question here says, is extractables testing even helpful in devices with limited exposure? In other words, can the results be used to waive irritation, sensitization, acute systemic toxicity, or material-mediated pyrogenicity?
That's a great question, and that's exactly my driving point on determining how much material characterization you're going to do. For a limited device, our concerns aren't really anything that can be answered with extractable leachables testing, where we can't get cytotoxicity information, sensitization, or irritation typically from that type of testing, because it's based on what's publicly available on the compounds identified, which we don't always understand those interactions. So we do feel like for a device like that, we would not recommend extractable leachables testing. We would simply recommend identifying what your materials are and stating that the use of those materials, the risk of the use of those materials will be assessed through biocompatibility testing.
All right, thank you, Audrey. We have time to sneak one more question in here. Our last question says, is the ISO 18562 accepted by the FDA for submission of a critical care ventilator, or is it necessary to use ISO 10993?
So this is a great example of one of those vertical standards. 18562 is more, specifically for breathing gas pathway devices. So the FDA has put on their website of acceptable standards that they do accept parts of, so 18562 is a 4-part standard series, and they have put the parts that they accept or do not accept from those standards on their website. So I would say that the FDA will accept 18562 testing, but you need to ensure you're understanding the parts that they're not accepting. And we've actually worked directly with a client as FDA was making this decision. So we've had several conversations and do know the specific testing requirements that they're looking for. But yes, you have to actually sometimes use both. So it depends on the contact with the patient. But if it's just the breathing gas pathway, we'll go to 18562. If there's some combination of both, then we can use both standards.
All right. Well, thank you, Audrey. Thank you, Thor. And thank you, Annelies, for a great Q&A session. That is all the time that we have for questions for today. Once again, if we did not get to your question today, someone will get back to you after the program is over. And within the next 24 hours, you will receive a personalized follow-up email with details and a link to today's presentation on demand. Please feel free to invite your colleagues and peers who may not have been available to listen to today's event. This webinar is copyright 2018 by UBM. The presentation materials are owned by or copyright by Nelson Laboratories, and the individual speakers are solely responsible for their content and opinions. On behalf of our guests, I'm Chris Keach. Once again, we'd like to thank you for attending this week's course, and we hope you'll join us again.